Okay, David was talking a little bit about metagenomics. He's the only one who's talking about metagenomics in this session, I think, but tomorrow there will be a session on metagenomics, both in clinical settings and in food, tomorrow morning, and I will encourage you to go to that one. But I'm going to give you an update of where we are and what our plans are for using whole genome sequencing in public health part of food safety in this country. And my title is a little bit provoking: is whole genome sequencing really replacing traditional microbiology? Well, traditional microbiology, this is basically a patient report of a sample, and those of you who can see it will be able to see that it's actually the STEC that caused the big outbreak of hemolytic uremic syndrome in Germany a couple of years ago. But in order to get all the information that we need in public health to act, we had to do a lot of things. First of all, we need to identify it as an E. coli using biochemicals. We need to do traditional serotyping, O and H agglutination. And in order to characterize a limited number of virulence factors, well, we had to do at least 2 PCRs and some IFLP. And in order to do MLST, we have to do 7 PCRs and sequence the products. And if we also wanted to include the resistance, well, we had to do disk diffusion or broth microdilution as we are using. And all this would take us up to about 2 weeks in order to get all the results. And in addition, if we wanted to subtype it, well, what we did back then, and what we're still doing actually, is doing PFGE. Last year, I presented some data on our initial implementation of whole genome sequencing for real-time surveillance of listeriosis, which was a project that we did together with FDA, NCBI, the Public Health Laboratories, and APHL. And it was a proof of concept study, and actually we proved that it worked. It is possible to perform whole genome sequencing in real time. It's definitely cost efficient to do it. And the big benefit is that we have unheard discrimination and precision. We can differentiate between epidemiologically unrelated isolates that have the same PFG pattern, and we can link case patients that are infected with different PFG patterns to the same single source outbreak. And that way we could refine outbreak case definitions. And also by comparing with the whole genome sequence of isolates from the food sources, we can increase the confidence in links between the clinical and the food sources. And then suddenly we are also able better to link historic case patients with current outbreaks. I'm not going to talk anything more about outbreaks and outbreak investigations. There are others who are going to do that here. So we are going to move beyond subtyping, and that is what I'm going to talk about now. At CDC, what we are doing is we have received some money from the Congress in the so-called Advanced Molecular Detection Initiative, which is a 5-year budget initiative that started back in 2014. So we are in the 2nd year now with an initial investment of $30 million, and we hope and expect to receive $30 million for the next, or for a full 5-year period. We received it also this year, and hopefully it's going to continue. And one of the projects that are being funded is something that's called Transforming Public Health Microbiology, PulseNet, and Beyond. And that is a project that I'm in charge of. And what we want to do there is basically to replace traditional microbiology in the public health laboratories in this country with whole genome sequencing for the characterization, the full characterization of foodborne pathogens. That way we can consolidate all the many workflows into just one single efficient whole genome sequencing workflow. We will be able to identify serotype, virulence profile, test the antimicrobial resistance markers, detect plasmids, and also do subtyping instead of doing multiple different tests, we'll just do the sequencing and then let the computers do the rest. When we succeed with that, and I'm not saying if, I'm saying when, because it is going to happen, then we will see a changing role between CDC and the public health laboratories out in the States. Out in the States, they will be doing much more. They're going to isolate the organism, they will sequence them, and then they will perform the routine analysis of the whole genome sequencing data from their own jurisdiction, but both for the use in their local surveillance, but also for the national surveillance. And what will be left for CDC? Well, we will not be out of work because we will have to do some data management and analyze the data, at least all the aggregated data at the national level. We have to produce search capacity for sequencing. We need to help with troubleshooting of the sequencing, and we need to keep the national organism-specific subject matter expertise, and we cannot give up on all the traditional microbiology. Somebody needs to be able to do that, so we will be some kind of center for classical microbiology, and we need that because we need to characterize new or emerging pathogens, also phenotypically, and we will need to also monitor some of the phenotypic characteristics that we're trying to predict from the genotype, because not all genes are expressed, and if they're not expressed, well, we need to figure out what is the correlation. Is there any change in the correlation, especially with antimicrobial resistance? And we will be able to integrate the laboratory and the epidemiology much more efficient this way. So the path to that, well, we have to build the capacity out in the States, and by ourselves also. We need to develop the protocols and the analytical platforms that we are going to use. We are also doing some clinical testing where the results are going to end up in a patient file, and that has to be certified according to the CLIA regulations. We need to validate everything that we are developing and using both at CDC but also by the external users that are going to use them in the end. And then, of course, we need to establish a quality assurance system without which none of this is going to work. So where are we right now? Things are moving fast. Last year, I don't think we had capacity for whole genome sequencing in more or less than 10 laboratories, and much thanks to FDA and the Genome Tracker project that has put a number of sequences in a number of public health laboratories. But today, there is capacity in 26 public health laboratories. And we have begun the external validation of the PulseNet Listeria whole genome sequencing protocols, including the subtyping and identification in 10 of those laboratories at the moment. And we're in the process of validating everything that we do with Listeria for whole genome sequencing for CLIA right now. We are developing, or we are in the later phase of developing and validating the PulseNet protocols for Zika-toxin-producing E. coli and Campylobacter ceae, and we expect that that is going to be moved out to the States within a few months and be tested out there. And we've just begun the development of databases for Salmonella. This is an enormous project, and we certainly cannot do that alone. And there are a couple of reasons why we can't do that. We do not have the capacity or the knowledge to make this transformation all by ourselves. And what we do, because food doesn't respect any borders, we need to be able to compare results in different countries and in different laboratories. So everything we do needs to be synchronized with what is happening in other parts of the world. So we are really doing this. This is one of the things that we have developed ourselves. This is the tool that we are using for identification of bacterial strains now to the species level. We're using a concept called average nucleotide identity, or ANI, which basically is a similarity measure of nucleotide identity between homologous regions between two isolates. And it's a very robust way to identify the species of an isolate if you have some good reference strains by whole genome sequencing. And the cutoff limit for species identity is more than 95% similarity. And just to show you how it works with Listeria, yeah, you can see it. Here is some ANI values for strains of the different Listeria species and ANI values for comparisons between the different species. And you can see there is fairly good discrimination between the different species. All the species is basically has ANI values above 95%, and then there is big gap, and then between species of the same genus is somewhere between 90% and 92%, something like that. And it works for this, and it will work also when we compare with strains of different genera. Then we are working with, of course, with a lot of international partners. And one of the international partners we are working with is a Danish partner from the Danish Technical University, the Center for Genomic Epidemiology, who has built a system of databases and a number of tools that you can use one-on-one and predict phenotypes or profiles from the whole genome sequence. One of the tools that we are implementing is serotyping of E. coli from the sequence. And this is a tremendous step forward because doing traditional serotyping of E. coli is cumbersome, extremely expensive. Using this tool, we can do the prediction of the serotype, which is based on the IAFP genes and the FLIC genes. So fully compatible with traditional serotyping, and we can do it just in a few days that it takes to do the whole genome sequencing. And also for E. coli, we are working on also detecting the virulence genes, and the Center for Genomic Epidemiology, together with Statens Serum Institute in Copenhagen, has also developed a tool that can be used for that, including the Shiga-toxin genes and all the associated genes that determines the virulence of E. coli. They have also developed a tool where we can detect plasmids and characterize the plasmids. And for Salmonella, we don't use the tool from this group, but we are working with the University of Georgia, where Xiangyu Deng is working. He used to be a postdoc with us, and he was working on molecular serotyping of Salmonella, and he has refined it when he came to University of Georgia, and now he has launched a tool that you can use on the web to serotype Salmonella, also using direct correlation to the Kaufman-White scheme, sequencing the IFP genes and the FLIC and FLJB genes. It doesn't cover all the serotypes, but it covers most of the important human serotypes at least. And finally, I'll speak a little bit about the susceptibility testing that we can do by using sequencing. We need to understand that, or you need to understand that it's not susceptibility testing per se, but it's rather detection of resistance markers or genes. And by these tools, we are not detecting mutations, but we are detecting resistance genes. And there are actually a number of tools out there on the web that we can use to extract resistance markers. But the one that we have chosen to work with is the RES finder, also from the Center for Genomic Epidemiology. As I also mentioned earlier, the resistance markers are not always expressed, so we have to validate this against the phenotypic data, and in production we'll have to monitor the correlation continuously. So we will need to establish, when we implement this routinely, we'll also need to implement a sentinel system where we are continuously correlating the phenotypic antimicrobial resistance with the genotype. And of course, because only the known resistance markers are going to be detected, we also need the sentinel phenotypic surveillance in order to be able to detect the new resistances. And as I said, we are using the race finder from the Center for Genomic Epidemiology. And we have tested it on Salmonella in the NARMS group in collaboration with FDA and USDA. And we've tested it on about 200 strains. And as you can see, the performance is remarkable good. The sensitivity is more than 98%, and the specificity is just as good. And of course, the positive predictive value, negative predictive value are excellent. And actually, if you compare this with the traditional phenotypic reproducibility of those antimicrobial resistance markers, this is actually probably better than the reproducibility of a traditional phenotypic antimicrobial resistance testing. So this is pretty good. But still, we will need to do the correlation between the phenotype and the genotype on a continual basis in order to make sure we don't overlook anything, any new resistance markers that we don't want to overlook. So everything I've talked about right now is tools that you can use and you can do at one at a time on the web. But in public health, we want to do everything at the same time. Everything that's relevant to one organism, we want to do that at the same time. And not having to log onto a website and then do one thing and then log onto another site, do the other thing, and so on. We want to do everything in the same workflow at the same time with just a push of a button. And what we are doing basically is we are establishing a system that is based on the PulseNet routine software actually that we've been using for 16 years. Well, it can also analyze whole genome sequence data now. So everything can be controlled by the end user, which is in the middle, up there on the top. They will start the sequencing, and once the sequences are out there, they will basically automatically clean the data and extract the data that we need to use. And this is it. The genus species information, serotype information, pathotype information, virulence information, resistance information, and all the subtyping information, and put it up to the end user where you can store it easily and in a much more simplified way than when you're storing the sequences, which needs to be in some of the big capacity databases. And another advantage of this format is it's very easy to analyze. You can analyze the data on a standard desktop. And once you have done whatever you want to do with it, you store the data in a public health database, or if you have to provide a response to a customer who provided you with the strains, well, then you just send it out with your LIMS system. I want to mention, ideally, right now, this function to extract all this data is available on some servers at CDC. But if this is going to be working on the international scale and also for the industry in this country, then it has to be publicly available, and we need to have those tools out in public domain. And yeah, I hope we can come up with a way to do that. Ideally, we should put that up in the NCBI if we can. So basically, what we want to do is if we get an E. coli like the German strain again, we will just sequence it, and then by using the different tools in one workflow, we'll get the species from the ANI that we have developed, the serotype using the serotype finder, the pathotype and the virulence profile using the virulence finder, the sequence type using the MLST, 7-gene MLST extraction from the sequences, and then the antimicrobial resistance genes using the ResFinder tool. And then, of course, we will need to explain what it is that's going on, but that is hopefully not a big problem. And this is just a projected timeline for how we're going to validate all this. It says it's projected whole genome MLST database validation and deployment, but it is basically all the tools I've been talking about that needs to be validated and deployed. And we are right here now with the Listeria. We've just started the external validation, but by the end of this year, it should be fully validated and can be implemented in the states that have the sequencing capacity. With the Campylobacter and the Zika tools, we are just about to start the external validation, and we expect also to be done with that by the end of the year or the beginning of next year. And we've just started with Salmonella, the development of the database and the tools, and then next year we'll do the pilot and then we'll implement it. And after that, all the other foodborne bacterial pathogens will follow. One of the first ones to follow next year would be the Vibrios, not just cholera, but also Parahemolyticus and Vulnificus and all the other important Vibrio species. And with that, I'll just stop. I have so many acknowledgements that I'm not going to mention anyone because I don't want to forget anyone. Thank you.